All right. So thank you everyone that's here so far to attend our sessions this afternoon or morning or night, I guess, depending on where you're located. Our first talk today is going to be from Lara Clemmens, who's a postdoc with DILIsim Services, and she'll be talking about some aspects of mathematical modeling in industry, kind of aiming our models towards advancing drug development, but some of the skills that are necessary to do that. So with that, I'll turn things over to Lara.
Thanks. Yeah, so today I'm going to talk a little bit about mathematical modeling in industry, and specifically what we do here at DILIsim to build models to advance drug development. So a little bit about DILIsim Services. DILIsim Services is a Simulations Plus company, and Simulations Plus develops models and simulation software designed to support drug development through the entire discovery process. So we go from discovery to clinical development, research, regulatory submissions, the whole 9 yards. Specifically here at DILIsim, we use quantitative systems pharmacology and quantitative systems toxicology to support the pharmaceutical industry in a variety of ways, which I will talk about in a couple of minutes. And we've developed numerous modeling software platforms to support this development we're very focused on safe and effective drug therapies. And so today I'll give you a brief introduction to DILIsim, and if you stick around, you can hear Nadir talk about RENAsim, but we also have several other modeling platforms which are available, including IPFSim, which was just released today. So what is QSP modeling, and what goes into the design of something that would be a QSP model? So QSP models are designed to characterize biological systems, and they represent lots of different details. They can include things like the disease pathophysiology. So this is the features of your biological system that would be different in a disease state or would be important for representing a particular disease. They can also include drug pharmacology, which is thinking about how the drug impacts your body, or depending on what model you're trying to build, a specific organ. And then they can also include things like the pathways of toxicity. So importantly, this is things like how a drug might impact a particular cell type and disrupt some of its standard mechanisms of function. So we often think about how mitochondria function and how drugs might disrupt that. We can also think about how you might end up with a buildup of reactive oxygen species, and all of these things could be included in a QSP model. And so typically, these types of models depict interactions between drugs and the biological system of interest. And so again, I'll talk a little bit today about DILIsim specifically, and that is looking at how drugs influence the liver specifically. And so here at DILIsim, we've developed our software by using a large system of ODEs. And so these systems are treating all of our biological compartments as well-mixed tanks. And then depending on what aspect of the biology we want to get into, we have very mechanistic representations of the relevant pieces. And we combine these with both in vitro and in vivo publicly available literature to parameterize our model and make sure that this is consistent with observed behaviors. And all of these models at DILIsim are currently designed in the MATLAB environment, but we've also been investigating how C++ and Julia might be used to improve our simulation speeds, particularly for a couple of our other platforms. And importantly, one of our latest release of DILIsim, DILIsim X, will be released using a C++ platform. So this is some of the buildup of the design, but what is the outcome of building such a gigantic model? So QSP or QST models can answer questions related to key mechanistic or physiologic aspects and effects on the body, as well as understanding areas of uncertainty. So things that we think about a lot are predicting clinical efficacy or hepatotoxic risk or risk of liver injury. And we also think about identifying the primary drivers of this efficacy or toxicity. So thinking about, can we identify with a particular drug, if it maybe induces some amount of liver toxicity, can we identify what we think the main reason for that actually is? And then we can also use these to predict exposure ranges for efficacy or safety of new therapeutics, and comparing these new drugs to profiles of other standards of care. Other ways that QSP models can be really helpful is that in development, you might identify areas where you really need more data to understand all of the phenomenon occurring in that particular submodel. And so this is one way that you can start to build a model, and you may identify areas that would be really critical for understanding how a drug might affect or induce a response in a particular system, and that can guide future experiments to yield more insightful data. And the importance of these QSP models has really gained a lot of recognition in the past several years. And so these are widely recognized within both the pharmaceutical industry and the regulatory agencies as being really important for being able to explore target feasibility, look at efficacy and safety, dose optimization, and just generally improve the entire pipeline. And so these have been increasingly used to inform decision-making at all parts of the drug development pipeline. Okay, so a little bit about developing a model at DILIsim and some of the skills that we would use to do so. So first, let's talk about what one of our models would actually look like. So the map that I have up on the slide here is looking at a broad overview of some of the ideas that we capture in our DILIsim platform. And so you can see up at the top here, we have the drug metabolism and distribution. And so we look at how much drug actually reaches the liver and that the liver is exposed to. And then we can think about different pieces of the liver that will be impacted by this drug. So for instance, we have some of the liver adjacent phenomenon, such as we have cholangiocytes, the bile duct cells, and the buildup of bile acids in the model. And we also have phenomenon such as lipotoxicity and building up reactive oxygen species and looking at mitochondrial dysfunction. And we have all of these pieces that are leading to effects on the hepatocytes or the liver cells, and whether or not they are surviving the exposure to the drug, or whether they maybe are seeing damage or liver injury. And then additionally, we also include other associated liver cells, for instance, innate immune cells that are resident in the liver or recruited to the liver in response to any liver damage that might occur, and how that feeds back on this entire cycle. And then all of this accumulates in classic biomarkers, and these are signals that can be measured in the clinic and are used to make clinical decisions about whether or not they're seeing liver injury in a particular patient, so that we can start from exposure and then work our way down to what we would predict the clinical outcome would be for this particular patient and drug and the entire system. And so this whole process is involves a lot of pieces. And so how do we build up these pieces? And what are some of the tools and the skills that we use to develop this entire model? So for any new model or submodel, the main thing is identifying where you're going to start. What is the need that drives the creation of that submodel? And so you are identifying any unmet clinical need or areas of active clinical development. And once you've identified an area that really needs to be developed, then the next key piece is understanding all of the biological underpinnings and limitations that would go into building out the necessary pieces of the model to explore that effect. So for instance, I'm talking about this in terms of the liver, so we certainly need to understand liver physiology and sometimes surrounding organ physiology, but then down a scale from the liver, we also need to understand the particular cell types that are in the liver and how those interact with each other. And then within the cell, we need to look at the different mechanisms that are happening there of, they're releasing signals to other cells, they're receiving signals, they're creating energy, they're responding to lipids. There's a lot going on. And all of those pieces are critical to understanding both the development of the model and then what the effects coming out of it will be. And so going through each of those pieces, and then additionally thinking about some of the extra things that can happen. So we often think about the biophysics happening. So for instance, when we start talking about the kidney later, then we also start to think about things like, well, how does fluid flow affect some of the drug delivery? How does all of these small pieces come together into this large system? And so we also have like chemical reaction networks of different things being metabolized. And then importantly, we're taking all of the in vitro and in vivo data that we can to try and understand what behaviors we need to capture and what the important outcomes that we would expect to see are. And so once we've kind of gotten an overview of all of these different pieces, then we need to decide, okay, what are the critical pieces out of all of that biology that we absolutely have to include in our model, or that perhaps we don't need to include to replicate any phenomenon and make our predictions. And then within that, what is the best style for modeling that particular feature? And then this leads us straight into the main modeling and software development part of actually sitting down and designing the ODEs and other models and coding and debugging all of this large system, and then doing a lot of parameter optimization of finding the available data for a given parameter or trying to restrict parameter ranges and developing tools to improve how we optimize all of these different model pieces. And so there's a lot of different pieces that go into this. And then after you've done all of that, you've probably learned something new, and or other changes in the field have occurred, or there are other people getting really excited about something new going on, and you can see that you might want to make changes or expand or add new features, and therefore we continue the process. And so these are just some of the main key tools that we use to develop this large system. And clearly there are other things that go into this. So for instance, I said that we were developing this system that will look at how a drug is affecting a particular person, but obviously not all people are the same. And we all know that in a mathematical system, where you start is really important. And so we think a lot about where is the starting point of our system, what does that physiology look like for a given person, and what happens when we change that. And so we think a lot about the starting point and whether or not there are specific variables that should be adjusted, or we look at a large distribution of different possible places that that variable might sit at a healthy human being. Or depending on the disease state, that disease might modify that baseline level, and we would need to take that into account for seeing how that would affect everything downstream. And so we develop what are called simulation populations, SimPops, using alternate starting parameter values, and we can compare these to clinical data sets to make sure that we're representing an appropriate population, and then see how that affects steady states and the dynamics of the system. And then importantly, when we're developing a lot of submodels, we try to do a lot of sensitivity analysis to try and examine when we build a model, where are the places that if you push it in one direction, how does that affect the response, to make sure that our model is really robust, and that when we combine it with the other pieces, that we are have confidence in our outcomes. And this can also lead to new areas of interest of, if you push it in one direction, if that's a physiologically relevant direction, does that lead to new responses? And what does that say? And what kind of predictions can we make there? And then clearly all of this is this huge, gigantic model. And so you are not alone doing this. We are a very collaborative group. And so we build these models with a very interdisciplinary team here at DILIsim. And so you're working in a group to build a particular submodel, but then you're building a submodel that's going in with the larger model at hand, working with other groups to make sure everything is integrating well. And then we also partner with experimental groups for data collection to get data to verify what we expect to happen and to try and parameterize our models to reflect reality. And then clearly, since you have so many people, you do a lot of communication. And so we do a lot of internal and external presentations and posters and client reports and publications to communicate our science to the rest of the world, because that's how sharing science works. And so all of these are really key components to developing this model and to making sure that we have robust responses and that it reflects reality, and then providing the results to either our clients or the public as appropriate. So one thing that you might wonder with all of this is, there are so many pieces here. How do we decide which pieces to start to include, either in developing a novel model or developing a new submodel? And so here's just a few of the things that are important for our company in deciding where to put our time. One of them is the DILIsim initiative. This is a public-private partnership, which includes many pharmaceutical companies. And they're all able to put their expertise and opinions towards guiding the areas of model development for both DILIsim and now RENAsim. Additionally, we can develop or modify models for proprietary client projects. So our clients will identify new diseases or organs of interest or new therapeutics that they're interested in us developing or modifying models to help them with their particular question. And so that can push a particular model in a particular direction for that client. We also will have cross-platform updates. I mentioned that we have a lot of different platforms, and some of these have overlapping concepts. And so for a particular platform, we might need a specific submodel for that platform. And so we'll develop it for that particular tissue or the disease represented in that platform. And then some of these, as they're developed, will be applicable to yet other platforms. And so one of the classic examples is the construction of the innate immune response and the different cell type structures that are needed for one model that might also be useful for a different model, and different cytokines that can go into these as well. And so we are constantly improving each of our platforms based on things that we learned in other systems. We also build models based on grants and partnerships, based on holes in the field, so that we can constantly try and develop novel models that will be useful. And then, of course, as new science emerges, we try and update our models to reflect the new knowledge in the field. Okay, so just to summarize, DILIsim Services uses mathematical modeling tools to develop mechanistic representations of diseases or of drug-induced organ injury. And we work in a very interdisciplinary collaborative environment to build these novel models and to expand our existing models. And so I talked a lot about how the different mathematical tools that we use are used at DILIsim, but we are a very interdisciplinary team. And so our team is made up of mathematicians and engineers and life scientists. So it's a very diverse group of scientists here. And so DILIsim uses these QSP and QST models to address questions of critical value to pharmaceutical companies. So for instance, we do a lot of predicting hepatotoxic risk, investigating mechanistic causes for the observed toxicity, and predicting safety and exposure ranges for new therapeutics and comparing to existing drugs. But more than just at DILIsim, including at our larger company, mathematical modeling is really benefiting the pharmaceutical industry in a lot of different ways, including being able to predict exposure starting from just a chemical structure, and then again looking at efficacy and safety margins. And the real power of mathematical modeling is gaining a lot of recognition both by companies and with regulatory agencies. And so this has been a very, very dynamic and growing field. And so just to introduce you to our team here at DILIsim, this is our team. You can see the people marked with little red boxes are the group of us here at SMB. And if you would like to learn more about what we do, then stick around and talk to us at our booth, and stick around and hear Nadir introduce DILIsim. Thank you.
Great, thanks for that talk and that overview of DILIsim and QSP modeling, Lara. I actually think we'll park here for a minute and see if anyone has any questions about DILIsim in general or any of the topics that Lara has discussed. Feel free to unmute yourselves and ask, or type into the chat box if you'd prefer that.
Hi, so I'm Kevin. I actually have one question.
Great.
So the question I have is actually for members in the, I guess, in the project team in general, are most of the members being assigned, like, say, multiple sort of projects to concurrent work on, or they are more, like, say, focus on like a single aspect or like a single subject before moving on to the different topic, for instance?
Yeah, that's a great question. So it depends a little bit on background and expertise as to what people tend to focus on specifically, but we have a lot of projects going on and people get to work on a lot of different things. And so, you might focus on something in particular. I spend a lot of time thinking about the immune response in particular. So I spend a lot of time developing the immune submodels, but I've worked on a lot of different projects within the team as well, and so there's a lot of diversity, so you never get bored.
So it is like more like I would say kind of like a case by case scenario. So depending on your, I guess, expertise. I see.
Yeah, I think that's true.
I see. Thank you.
Hi, my name is Derek. I have a question too, if I could.
Yeah.
So my question was about the transition that you're doing from sort of away from MATLAB to either C++ and Julia. I guess two questions. So one, why are you moving away from MATLAB? And then sort of what standard for C++ are you using? And I guess what are your experiences with Julia? So three questions.
Okay. Yeah, those are all great questions, and I don't know that I will be able to answer all of them for you, but I will certainly try. So we've done a lot of development in MATLAB, and that has worked really, really well, and it's very user-friendly, particularly for people of different backgrounds to come in and be able to develop in that ecosystem has been really nice. That being said, some of our models have expanded quite a bit, and so in order to make them efficient for our clients to be able to run themselves, we do license the software to other people that they should be able to run in a reasonable time frame. Some of the time frames that we're investigating are really long, and so in order to keep the simulation time down, one of the things that we've been looking at is the feasibility of moving into a different programming language and how that would improve our simulation time, such that when we run, for instance, a simulated population where we're running hundreds of simulations, how does that affect our ability to produce results in a timely fashion? I came in a little later to the C++ development, so it's possible somebody else can answer questions about the exact C++ setup that we're developing there. But our latest release of DILIsim will be in C++-based platform, and that has been really nice for improving our simulation speeds. And one of our other platforms, they've been investigating looking into developing it in Julia. And if you'd like to know more about that, then we can talk a little bit more about that later. I don't have all of the details, but it's possible one of my colleagues would.
Thanks very much.
Great questions. Anyone else? Any other questions lingering out there? All right. If not, we'll thank Lara again for a great presentation.